Okay, so this is part one of our volatility lab. We're going to be comparing three different solutions. One is tap water, we have methanol, as well as we have acetone. In this portion of the lab, what we're going to do is we're going to use these droppers here. We're going to drop a droplet or two here on this glass plate here, start the stopwatch to actually just qualitatively compare which one seems to evaporate more quickly or less quickly as a measure of the intermolecular forces. So I'm going to just start off here with the water solution here. So just a droplet of water, holds its shape very well here. We have methanol, I'm just going to leave on top. And then we're going to have our acetone solution. Okay. I'm going to start my stopwatch here and we're going to make a qualitative comparison, which one seems to evaporate most quickly. Hopefully it's clear on the video here, but if not, I'll be a judge of sort of when it looks like these droplets are gone. Although I did try to have just one drop of each of these solutions, both methanol and acetone, they have trouble sort of holding into these droppers as sort of a qualitative comparison of the intermolecular forces here. Just give you an observation of when it looks like these solutions have disappeared. If the solutions have much stronger intermolecular forces, the sort of liquid-to-liquid strength holds itself together better, we would expect they would evaporate a lot slower. So. At this point here, we still have acetone, methanol. Our water droplet is still holding its shape very well. They use acetone in a lot of nail polish removers here, so if you accidentally got some polish remover on yourself here, you probably felt a very cooling effect here. I don't know if it's clear on the video here, but this acetone puddle here is basically evaporated away in about 1 minute and 5 seconds or so. So that's, again, a qualitative comparison here. Maybe liquid to liquid, it doesn't hold itself together very strongly, so again, it evaporated off quite quickly. In biology, you would call that evaporative cooling. So let's just wait on the other two here. We have our methanol solution. Although both the methanol and the acetone solution was a much larger puddle, a much larger pool to begin with, we can just qualitatively compare how long it's taking for these droplets to evaporate off. You're starting to see some changes in the methanol puddle here. There's a little bit of a clear spot in the middle of the droplet here. Looks like it's just about evaporated here, although the water is pretty much keeping its shape, holding its shape there. Again, it's slightly not fair for comparison because the droplet size were a little bit different. The water drop I can pretty much keep to a single drop. Methanol and acetone were maybe 2 or 3 drops altogether. But even qualitatively, you can make this comparison here. So we just have a sort of last little rim of methanol here just evaporating away. We're about 3 minutes now. Whenever we record times, even though our stopwatch has sort of minutes and seconds there, we would usually just convert everything to seconds. So if you did want to do a rate calculation later on, when you have seconds, you can just divide by seconds here. So. Just waiting for this last little bit of methanol to evaporate off. Okay, the acetone had long gone. definitely a lot of the methanol had evaporated off. No real change you can see in the water droplet here. So again, we can just make a qualitative comparison. So I'd say at this point here, most of the methanol is gone. Yes, there's the two smallest droplets here. This is sort of experimental error here, so you can talk about why is it that there's still a little bit of residual left over. But again, essentially no change has happened to this water droplet. Just leave it for another minute or so, see if there's any other dramatic difference between these times. All right, so we're going to end it off there. You can stand around for a lot longer. We can just say more than 10 minutes for this water solution here. Perhaps the methanol solution actually had traces of water left over in the beaker. Maybe that's actually water. That could be a reason why those two last little droplets are still hanging on there. Okay. But at least you get a qualitative comparison between these solutions. Thanks, guys.